So, student, welcome. In last lecture, we have learned about the replication of DNA, in which we have seen semi-conservative type of replication. Now, why it is called as semi-conservative, we have seen because when a new DNA is formed, in that new DNA, one strand is of parent strand and other strand is a newly formed strand. So, in that particular daughter DNA, the parent strand is conserved. Means half the structure of daughter DNA is old one and half the structure is new one. That is a semi. And conserve, what is conserved there? A parent DNA strand is conserved there. One of the parent DNA strand is conserved there. And so this type of replication is known as what? Semi-conservative type of replication, in which we have seen the whole process of DNA replication. Now, this DNA replication process have been experimentally proved by. This is experimentally proved by Meselson and stalh experiment. By which method? By which technique? Equilibrium density gradient method. You know equilibrium, what is equilibrium? Density gradient, if you see what happens in this experiment, they use cesium chloride. Cesium chloride, 6 molar of cesium chloride solution was used for obtaining equilibrium density gradient. What happens? Centrifugation. In this method, for getting density gradient band, they use centrifugation method. In this centrifugation method, when a particular substance is centrifuged, what happens? The particles, that particular solution in which very particles are present, they are separated by their density. Heavy particles settle down at base, lighter particle at higher level in a test tube, or in a particular container in which it is taken, and centrifugal is a machine in which the mixture is completely mixture is spin and spin, and by the spinning method all these particles which are present in that particular solution get separated, and according to the density that particle forms a band. And that particular level at which the particles settle, same particles settle, that forms somewhat band. And that band is called, that particular is called as density gradient of that particular solution, particular particles present in that solution. So by this method, this Meselson and stalh experimentally proved that the DNA replication in eukaryotic organism is semi-conservative type of replication. And for the experiment, which material was used? They used N40, light not nitrogen. Isotopic N40. Then they use N50, heavy nitrogen. Then CSCl2, 6 molar of CSCl2, cesium chloride. Then centrifugal machine, centrifugal apparatus. So, in the experiment, particularly, they take this isotope of nitrogen 14 isotope, then N15, which are heavy nitrogen, and 6 molar of CSCl2, cesium chloride, and centrifugal apparatus they take. Now, and E. coli, which bacteria was taken for the experiment, E. coli, Escherichia coli. This bacteria was again taken by them. Now we will see how they do this experiment, how they perform this experiment. The E. coli was cultured in medium having N14. E. coli, first step was there. E. coli was cultured in medium having N14 nitrogen. N14 nitrogen, and a equilibrium density gradient was obtained from it, and equilibrium density gradient obtained by using 6 molar of CSCl2 solution. This was, and that band was recorded. Then in second step, what they do, E. coli was cultured in medium heavy with N15, heavy isotope of nitrogen. N15, which is denoted by N15. And band was recorded. Means these bacteria, E. coli, what they do, they culture it in the cultured medium, and this cultured medium contains what? N14. And due to using of N14, what will the result we get in that culture, means colonies of E. coli? The newly formed E. coli will have N14 in their DNA strand. N14 in their DNA strand. So, and the band was obtained when this was treated with what? 6 mol of CSCl2 in a test tube. They have taken it, and by centrifugal method, a band is obtained, and that band record is recorded. Then in second step, what they do? The E. coli was cultured in with which medium? N15. The culture medium with N15 was taken, and then in that culture medium the E. coli was grown separately, and by that method what will we get? The daughter E. coli which we get, or the colonies which is newly formed E. coli will have N15 nitrogen isotope in their DNA. And by equilibrium density gradient method, by centrifugation method, a band was recorded. Then we will see the next step. The N15 DNA will transfer to another medium or N15. Now what they do? They transfer the N15 DNA, the culture in which that E. coli was cultured, E. coli with N15. Such N15 DNA E. coli will transfer in the medium which contains N14 nitrogen. N15 E. coli, N15 DNA E. coli will transfer to medium having N14 nitrogen. Now, by this method, what will we get? Well, in the first generation, the density gradient band for N14 and N15 was obtained. Now, if we take this N15 DNA and N14 DNA, if there is a mixture, what will we get? We will get a DNA in which one strand is of N15, one strand is of N14. So in first generation, when this was done, a density gradient band is produced and it is recorded. And the band which we get was DNA was of N14, one strand of N14 and second strand of N15, means this hybrid strand. Now if you see, we have culture, we have transfer N15, we have transfer N15 E. coli in the medium having N14, and when the daughter DNA was obtained, it contains one strand of N15 and one strand of N14. Means parent strand was conserved in the daughter, and new strand was a form of N14. So there is a hybrid DNA which contains the nitrogen of different isotopes, nitrogen of different value or isotopes of N14 and N15. Now, in next step, second generation was obtained. In second generation, in second generation, what they get? Two density gradient bands were obtained. Two density gradient bands were obtained. Two density gradient bands obtained. One at N14, N15 position, and other at N14 position. One at N14, N15 position, and other at N14, N14 position. And from this, the Meselson and stalh proved that the DNA replication is semi-conservative type of replication. Now, I will explain this with the help of figure, these two steps. Suppose when N15 will transfer this N15 DNA, both strands of N15 will transfer to which material? N14 medium. Then after transferring this, what we get? One strand, two DNA strands where replication was there, and after replication, these both strands are of N15, N15. And after replication, what we get? This strand of N14 and this strand of N15. This strand of N15, N14, and this strand of N15. So hybrid one we get there what? The DNA consists of one parent DNA and one which the daughter DNA. By using the medium of N14, the DNA was of N14. So hybrid DNA we get, or a daughter DNA which we get a newly strand is formed, newly synthesized strand is formed, and one is retained from the parent. And from this, a band was recorded. Suppose this is a test tube in which centrifugation is there. The band was recorded of N15, and this N15 is of heavy. It is a heavy, and so heavy particles settled down near to the bottom of the test tube. So this band was formed at this position, at the near to the base. Then after taking by centrifugation of this material, a band was recorded, and this band recorded here. It is not near to the base. It is forward one. So this band was obtained, and this band was what? Of N14 and N15. N14 and N15 nitrogen DNA. And again, when they were for second generation, when it was taken, we can obtain here from this two DNA are formed, from this two DNAs are formed, from these two DNAs are formed. Here it is hybrid one, N14, N15, or N14 only, N14, N14. This is N14, N15. this is N14, N15, and this is N14, N14. Two are hybrid, two are not hybrid, they are normal one with light nitrogen, normal nitrogen, and in this step we get two bands. We get two bands. Now you have seen this is at near to the base, this is again forward to the base, near to the middle. Here this position here, and this position is completely same, and this is of what? N14 and N15. And here a new band is formed. Here this is a light nitrogen. Light nitrogen is at the upper side because the density gradient particles, heavy particles form bands here, lighter than there, here, lighter than there, here, lighter than there, here, lighter than there, here, most lighted up to the position at the test tube. So this is of N14 and N14. So by this centrifugation density gradient method or equilibrium density gradient method, in which centrifugation was held, bands were obtained, and from this they conclude that the DNA replication is of semi-conservative type of replication. So this is the stalh and Meselson experiment, and you should keep it in your mind that 6 mole of CsCl2 was used. What is density gradient? You should keep it in your mind, and which nitrogen isotopes were used. That you should take in your mind because they are very important for NEET also and CET also, N14 and N15. So such is the experiment of Meselson and stalh. Then in next lecture tomorrow we are going to see protein synthesis. We are going to learn about protein synthesis. In 2 to 3 minutes I will introduce that protein synthesis. Now if we see various activities in animals or living things are controlled by enzymes. And these enzymes are made up of what? Proteins. But you should keep in your mind that all enzymes are made up of proteins, but not all the proteins are enzymes. Enzymes are made up of protein, no doubt, but all the proteins which are present in living things are not enzymes. And we know the role of enzymes. They prove all the biological reactions which are going in our body are controlled by enzymes. They help this reaction, they carry out this reaction, and they are needed. And various functions in our body are enzyme related or protein control. Protein control we have seen in the cell structure last year, in which we have seen ribosomes. What is the second name of ribosome? They are the protein factories. They are the protein factory, and protein synthesis is most important. And we have also seen the proteins. Proteins are what? Proteins are made up of what? Amino acid. Amino acid chains are present, and that chains are called as what? Polypeptide chains. That chains are called as what? Polypeptide chain. And this polypeptide chain may consist of the same amino acid or different amino acids. This chain may consist 2 to 3 amino acids, or it may contain more than that, infinity. So according to that, the name of that particular substance also changes. So this protein synthesis process is directed by the DNA. Now, this is not directly done. This is not directly done because for protein synthesis, a coded language is coded on what? DNA strands. on codons. If we see, last year we have seen RNA and DNA. There are codons present. Or if you see the RNA structure, there are codons present, and there is a triplet of amino acids or triplet of bases which forms a codon, and one codon specifies certain amino acids. One codon specifies certain amino acids. So these amino acids or these proteins, they perform particular function. So from DNA, directly there is no DNA no protein synthesis. From DNA, mRNA coded this language. Codes this language. mRNA codes this language from DNA, and from this mRNA, then and there is formation of proteins, or there is formation of polypeptide. There is formation of polypeptide. First of all, when there is a formation of mRNA from DNA, that is called as what? Transcription. That is called as what? Transcription. And after formation of mRNA, from this mRNA, there is a protein synthesis, or there is formation of protein by which method? Translation method. And in next lecture, we are going to see this protein synthesis, in which first process we are going to learn, that is transcription. Transcription means to copy. Transcription means copy. What is copied here? The DNA. From DNA, the coded language for protein synthesis is copied on mRNA. And this mRNA is transferred in cytoplasm, and in cytoplasm it attaches to ribosome, and it directs the process of protein synthesis. So we will see the transcription in method in next lecture. And if you see, when from DNA mRNA is formed, mRNA can reverse this process. From mRNA, DNA is also formed. By which enzyme? Reverse transcriptase enzyme. By which enzyme? Reverse transcriptase enzyme. It is done in some certain viruses. Yes, if you see, retrovirus, virus of AIDS, HIV virus. So this HIV virus, in this core medium, there is presence of the enzyme reverse transcriptase, and due to this, from mRNA, DNA also can form. And in certain process, there is no formation of this, then only transcriptase enzyme plays its role. So transcription we will learn in next few lectures.